On August 9th, 2023, we gave a conference presentation at a meeting of the Center for Cellular Construction. The subject of the presentation was a cellular biology experiment going on at UCSF, but the presentation itself was also a kind of experiment. Instead of PowerPoint slides, we flipped through physical pages with our hands, including research papers with videos embedded in them. We explained the history of the field by reconstructing seminal projects as computational models, live on the table, and kept everything in context with a persistent timeline over on the wall. We demonstrated the current experiment also by building it live with our actual design tools, and showed the resulting data. After the presentation, the audience got to try these tools for themselves. This all took place in Realtalk, our environment for combining computation with real-world objects. In this video, I'll give a brief background on Realtalk, including past biotechnology projects as well as past presentations. Then I'll give a summary of each of the 3 parts of our conference presentation and show what was significant about them. And I'll talk about the session after the presentation, where audience members could then try it out for themselves. This meta presentation will be about 15 minutes, and that'll be followed by a recording of the full presentation itself, which is 70 minutes and assumes a more specialized audience. Realtalk is a computing environment, or operating system, for real people working together in the real world. There's no screens and no apps. Instead, people craft computational activities using real-world materials out in the open where everything is immediately accessible and everyone can learn from everyone else. For several years, we ran a community space called Dynamic Land, where 1,000 people of all kinds with different interests and backgrounds made hundreds of projects to communicate their own ideas. One of our community members was Shawn Douglas, who runs a DNA nanotechnology lab at UCSF. Both Shawn and the Dynamic Land team were very interested in how Realtalk could support the work of scientists, as well as scientific thinking for everyone. In 2017, Shawn brought some colleagues to Dynamic Land to build a tangible environment for designing recombinant DNA. We all created it together out on the table in a group discussion. In 2021, we prototyped a DNA CAD tool where each component of the program could be introduced one at a time and explored in conversation as we gradually built up the complete program to design a DNA nanostructure. In 2022, we prototyped a set of tools for biomolecular design. We could design molecules together by building scale models out on the table, explore ideas by hand drawing over computational renderings, and actually see what we were doing in the wet lab. And in 2023, one thing that happened was an opportunity to give a presentation. So Realtalk was intended from the beginning as a medium of communication for people to discuss ideas using computational models. And one way we wanted to use this medium was to give dynamic presentations. At Dynamic Land, there were a number of presentations using Realtalk, where the presenter dealt out programs and dynamic media instead of showing slides. Some presentations were for a seated audience, and other ones were more like tours around a space of computational models. We also brought Realtalk to places outside of Dynamic Land and gave live demonstrations. What we had never done before was to go to a conference and use Realtalk to give a presentation about something other than Realtalk itself, using Realtalk as the medium, but not the subject of the talk. In a typical conference presentation today, it's just a speaker alone on a stage with a big screen, so they can't make use of the entire space to show context or persistent peripheral information. The screen typically shows a sequence of slides, so when the next slide is shown, the last slide disappears and the audience can't refer back to anything. And the sequence of slides is fixed in advance, so the presentation has to stick to the script. The materials don't help the speaker with digressions or discussion, which are the reasons you'd actually want a live speaker. And typically the slides are just words and pictures, so the speaker isn't actually using the computer to compute anything. Despite that, the presentation software may be tens of millions of lines of code. That's an overwhelming amount of complexity, so most speakers can't change the program or program their own presentations. They're restricted to generic mass-market software packages. Our goal with this presentation was to begin to demonstrate what scientific talk could look like in a humane dynamic medium. The presentation took place at a conference of cellular engineers and synthetic biologists. There were about 80 people in the audience. We set up the space so Shawn and I could work on a table in the front of the room, and we had a small side table for our materials. Joanne was next to us holding a camera, and the camera feed was projected on the screen behind us so the audience could see a closer view of what we were doing on the table. We'd also set up Realtalk on a sidewall for a persistent dynamic timeline in the periphery. The presentation went well, but unfortunately it was not recorded, so what you'll be seeing here is a reenactment we did a week later in Shawn's office at UCSF. This talk has 3 parts. In the first part, Shawn introduces his lab's collaboration with Orion Weiner's lab. They're building tiny playgrounds for cells to crawl around on, in order to study how cells are able to crawl around. Instead of a PowerPoint deck, Shawn collected materials about the collaboration into a 3-ring binder, where he can freely flip through the pages, take them out as needed, spread them out, show them to us with his hands. Some of these pages are more like slides with textual prompts and figures. He also has research papers in the binder that he takes us through, and these papers have embedded media in them. By pointing a card directly at a figure in the original paper, it reveals the video for that figure, and he plays the video and scrubs around in the video as he talks about it. These videos were originally published in the paper's supplementary materials online, where they're not very easy to get to. Shawn implemented the program for attaching and playing videos in one page of Realtalk. Here's the entire program, and here Shawn is talking about how the program works as he points to each part of it. The binder and slides, of course, require 0 lines of code. They just physically exist. Shawn made this binder for this presentation, but in the months afterwards, there were many times when Shawn would pull out the binder in a meeting with colleagues or funders, and they would all go through the material together. This fit very naturally into the flow of the conversation, and the conversation could continue with the materials out on the table. This felt very different than earlier meetings where Shawn would sometimes pull out his laptop or phone in order to show presentation materials, and everyone would crowd around the screen. This always felt really awkward, and the device would get put away as soon as possible. So one notable thing about Realtalk presentations is that they seem to be continuous with conversations. The presentation materials become conversation materials in a very natural way. The cellular playgrounds described in the first part of the talk are built out of DNA nanostructures. That brings up the question of what are DNA nanostructures, and how did people ever figure out how to build structures out of DNA? So in the second part of the talk, I give a history of the field, going through the lives of some of the key people and the key papers that they published. I have a timeline over on the side wall, and on the table I have a stack of person cards. Each card has a person's information on it, and when the card is showing, we see the person's lifespan on the timeline and the institutions that they were at. There was a lot of cross-pollination among these people and their institutions, which is an interesting part of the story. I also have a stack of published papers. These were the seminal papers in the field, and as I put each paper on the table, its icon is added to the timeline. So as the talk proceeds, you can always see the entire sequence of papers so far in context. I can point a laser pointer at an icon to show thumbnails of the pages. I can also show the pages by physically flipping through the paper itself, and I do both of these throughout the talk. As we go through the sequence of papers, I want to show what was significant about each one. So instead of just talking about it or pointing at figures, I recreate, live on the table, the structure described in the paper. To do that, I use a set of cards for modeling DNA strands. I have a card that adds bases to a strand, a card that removes bases, a card that forms crossovers, cards that nick or ligate or recolor, and so on. About 6 double-sided cards. These cards don't know anything about the papers, they're just a generic toolkit. But of course, I made the cards for this talk, so they have what I need to express the structures from the papers. And each structure leads into the next one. We start with Alex Rich's paper about the discovery of hybridization, and then in Ned Siemens' paper, he's using hybridization to form different kinds of junctions, and talks about how an immobile junction with sticky ends could tile into a lattice. In Ned's later paper, we have the invention of the double antiparallel crossover, which is a crucial building block in DNA nanotechnology. And that's used in Paul Rotemann's paper to make large asymmetric shapes with a technique he called DNA origami. So as I present each paper, I'm building the structure it describes out of the structure built up for the previous paper. The last paper is from Peng Yin. He uses those antiparallel crossovers in a different way to make DNA bricks, and that's the technique that Shawn's lab is using in this project to make cellular playgrounds. Now these diagrams I'm forming on the table are 2-dimensional schematics, but they actually represent 3-dimensional structures made out of DNA double helices. So we want to be able to see what these things actually look like in 3 dimensions, and I have a couple ways of doing that. One is by rendering them in 3D on a piece of paper, but the way that you control this rendering is by arranging physical models on the table. So each of these plastic tubes is a scale model of a DNA helix, and as you position them on the table and spin them around, it's that scene on the table which is being rendered. And the viewpoint for the rendering is represented by this garbanzo bean can. That's just what I happened to have when I made this. The scene is rendered as if you're looking at it from the edge of the can, so you can fly in and out by moving the can. Because the tubes are clear, it's easy to see the helix in real life. You can see where all the nucleotides are even as they go around the back, and you can trace the helix with your finger. And the nucleotides are numbered, with the same numbers on the 2D schematic, in the 3D rendering, and printed on the tube, so you can make the correspondence between all 3 representations. You can also point at the schematic with a laser pointer to light up the corresponding nucleotide in the rendering. In these structures, it's important what angle the helices meet at. Here you can see that they form an L shape. That's much easier to see in the real-world scale model on the table than in the 3D rendering. So instead of just talking about this historical work, I'm presenting it by recreating it live as a computational model. One consequence is that it sets up for, later, guiding a student through using the same tools to recreate it for themselves, and thereby actually learning something. That happened after the presentation, and I'll get to that in a bit. Another consequence is that we can bring in programs to analyze the structure as we're building it. I don't show that in this talk, but we have little programs which will compute, for example, the binding energies of the duplexes and the overall stability of the molecule as you play with it. I think that if you're talking about a computational model while seeing a live simulation of its behavioral properties, you're less likely to be fooling yourself. Here's the implementation of this part of the presentation. This page draws the timeline. This half page is for interacting with the timeline. These two pages take care of drawing and manipulating the 2D representation on the table. This page notices when two strands are lined up so they can be paired as a helix, and this is the page and a half which renders the 3D scene using the tubes. So that's my presentation software. Except each card also has a little program on it to add bases or form crossovers or whatever it does, so there's between a couple lines to a dozen lines on each card, and those lines are currently not visible. So that's a violation of our principles, and we have some ideas for how programs on small cards in Realtalk could be more visible. The previous part of the talk culminated in explaining the DNA bricks technique. In this last part of the talk, Shawn uses a Realtalk-based toolkit to actually design and fabricate real DNA brick structures, which were used to get experimental data about cellular playgrounds. Shawn starts this part of the talk by pulling his toolkit out of a zipper pouch in the binder. So the binder contains both presentation materials and working tools. This tool is the voxel editor that Luke made for defining three-dimensional shapes. Here Shawn is placing a sphere into the scene by pointing into the page with a sphere card. He moves the sphere to the bottom of the space to truncate it into a hemisphere. Then he adds a second shape, a negative sphere, in order to hollow out the hemisphere into a shell. He's doing this by pointing at a front view of the space, but on a separate page we can also see a top view, and he can operate it on there as well. The shapes he's adding have parameters, such as their dimensions, which can be adjusted by turning a small card as if it were a knob. Here, Shawn picks up a composite shape into a token, which represents a test tube. He makes a couple more designs, and picks up each of them as well, so all three designs are on the table. He looks underneath the design by tumbling it with a laser pointer. Then he uses other tools to process these designs. This tool converts a voxel shape into a set of DNA sequences, following the DNA bricks technique. He picks up the generated set of DNA sequences into its own tube. So on the left is the geometry he specified, and what's shown on the right is the nanostructure that these DNA strands will fold into. The next step is to combine these DNA sequences into a format that's suitable for ordering from vendors and using in the wet lab. By placing them into this box, the tool generates an order and even estimates the cost. There are other tools not shown here that convert these designs into a step-by-step procedure for fabricating the structures in the wet lab. These shapes were, in fact, ordered and fabricated and tested with live cells by the students in the Weiner lab. Here, Shawn is pulling the results of the experiments out of the binder. These videos show the results with the fabricated nanostructures, as well as the results of the control experiment using an alternative method. And here are some plots analyzing these results. The team was satisfied that they could replicate the old behavior using the new method, and can now move forward with new designs. So instead of just showing pictures of the nanostructures and describing how they're built, Shawn is actually building them, live, as part of the presentation, so the audience can see where they came from. The algorithm for generating DNA sequences was not made visible here, but in future presentations, we intend to build the algorithms live as well. Here's the entire implementation of the voxel editor and DNA sequence generator. It's about 8 full pages. Luke likes to collect his kits on portable whiteboards that can be placed on the table or hung on the wall. The implementation of picking up designs and moving them around extends from a set of biomolecular design tools we had prototyped the previous year, where we would pick up proteins and DNA structures into these little test tube tokens. So for this DNA brick workflow, we're reusing 2 pages from this poster from last year. My favorite part of the presentation happened after the presentation. We'd set up Realtalk in another room, so after the presentation was over, audience members could then get their hands on what they had seen. One student came in and wanted to build a holiday junction. She had seen pictures of holiday junctions in textbooks where they were always drawn like this. And I think that representation is super confusing because that does not look like a crossover. So in the talk, I started by making an obvious parallel crossover, and then smoothly rearranged it with my hands into the textbook form. And that's what the student wanted to do as well. So I coached her a bit through using the cards, and she made her holiday junction on the table and played with it, and she was excited that holiday junctions finally made sense to her. To me, this brings up the question of what the purpose of a lecture presentation is in the first place. I believe that in many domains, a person doesn't learn very much from hearing someone else talk. They need to actually build something with their hands. So one possible use for a presentation is to demonstrate a set of tools and concepts which the audience then uses themselves with coaching to reconstruct the presenter's work. And you see this pattern in woodworking classes, for example. The students will watch the instructor demonstrate how to use a tool or how to cut a joint, and then the students will do it themselves, with the instructors coaching them individually as needed. Woodworking is knowledge that lives in the hands, so this can't work any other way. But this is not how scientific presentations work. At a conference, a presenter will talk about the experiment they did and show slides of their data, but the audience does not then recapitulate the experiment themselves, even in simulation. The audience does not explore and analyze the data themselves. They just listen. We believe that scientific presentations, as currently structured, prevent scientists from understanding each other's work as readily as they could. We're eager to use Realtalk to create environments where attending a presentation means actively reconstructing the presenter's work, getting the knowledge into everyone's hands. The presentation shown in this video is an early step towards this vision. Before we get started, I want to give a quick introduction to this computing environment so you kind of have some idea of what you're seeing. Realtalk, our operating system, is designed for real people working together in the real world. So instead of people being on laptops dragging around icons, people gather around tables and physical spaces and work with physical materials with their hands. And so in this environment, any physical object can be a computational object if you put a program on it. And programs themselves are physical things. So this, for example, is a program. And when it is down, it does what it does. So we can all gather around and look at this program. Anyone can read it. We can point to parts of it. We can talk about it, and anybody can change it right as it's running. We can make the program different, and now it's doing a different thing. So it's this very fluid form of computation that's designed for people working together and having discussions together. And to give you a brief little introduction to what that means, I'll show you a little bit of a project that Shawn and I were doing last year. So Shawn had a project going on in his lab that involved making a protein fusion. They wanted to splice two proteins together. And so if we imagine, how would we like to go about doing that? Well, maybe we have a card and we type in the name of one of the proteins onto the card, and it brings up the results from the protein data bank. And so this is the one that we want to work with, and so we can pick it up into one of these mock tubes. It's like a game piece in a board game. So now we have this protein here on the table, and I can use the laser pointer to spin it around and take a look at it and think about how we want to splice in our other protein. So the other protein that we're going to grab here is good friend GFP. And so pick up that one. And now we can think about where do we want to work with these things. So we can look for a good spot to bring it in. Maybe it's that little loop-de-loop right there. We want to bring it in into the host protein. And so in order to do that, first thing we want to do is open up the GFP there so we can loop it in. And I've got a little program to do that. This program is permute proteins. And when I put it down, it extends a little whisker pointing to the spot on the protein where we want to open it up. And it puts its result over here where I can pick it up into its own tube. So now we have two proteins, the old GFP and our new improved GFP. You can see we have the termini at the spot we want them. And I'm going to swap that into the scene. And now we want to splice that into the host protein. And I again have a little program to do that, splice proteins. Same sort of deal, extends a whisker to the spot that we want to splice it in. And I can pick up the results into a tube like that. So we can put all these things on the table and we can all gather around and take a look at it and discuss these things and make little programs that operate on them, all in this very fluid, conversational, communal manner. And that's just a little bit of what we did last year. We brought this system into the wet lab and we had test tubes which showed what was inside them. We looked at microscopy data, but that was all stuff from last year, and we're excited to show what we've been working on this year, meaning the last few weeks. So, Shawn, over to you.
So my lab has a collaboration with Orion Weiner's lab, and Orion studies cellular activation, shape, and movement. And so here's what his lab website looks like. And I thought it would be fun to collect our collaboration into a 3-ring binder. And so normally we would show PowerPoint slides, but here we can just kind of flip through this binder, and this has all the materials related to our collaboration. So so first I want to mention Orion has a nice team of scientists that he works with, and Mizuye Wu is a graduate student, and she's been leading the Orion side of our collaboration. And then Conlin and Raghu in my lab have been leading the DNA nanotech efforts. So as I mentioned, the Weiner lab studies cell shape and how that affects cellular decision making, and they use neutrophils as a model system. And so when you have a wound, like a cut or a burn or something, neutrophils will sense chemical cues and then migrate to the site of the wound and sterilize that site. And so when Orion presents this on slides, he usually shows a movie from a 2013 paper from Ron Germain's lab. And so why don't we just pull up that paper? And so this paper is not exactly about neutrophils responding to shape, but it does have a nice movie kind of buried in the supplementary material. So if I flip the page and I look at this paragraph here, there's a mention of a supplementary video number 2. And we would have to dig that up normally, but since this is a Realtalk object, we can actually attach media to including videos to this page. So, and then I have this little card here that I can use to point at that and pull up the video. So here's what Orion would normally show. So we have this is a mouse model, and what the researchers are going to do is use a laser to induce a wound site right here. It's a time series of minutes and seconds, so we can watch the progression if we play the video. And so we see this swarm of neutrophils, which are labeled with a green fluorescent protein. So we can see the cells. They all sense that wound and then now migrate to the site. And so this is just dramatic and really amazing what the cells are able to do here. And so we'll put this paper away and just moving forward. So next, just again channeling Orion. He likes to zoom in from this high level view of a lot of neutrophils and usually look closely at a single neutrophil. And for that, he uses another movie that was actually recorded in Dyke Mullins' lab, and it was part of this famous lattice light sheet microscopy paper. All the friends at Woods Hole kind of brought all their different cellular systems and contributed to sort of the testing of Eric Betzig's lattice light sheet microscope, and then ended up all as co-authors on this paper. And so a lot of cool movies in this paper. We're gonna skip over those and then pull up the one that Orion usually shows. And this is a single neutrophil crawling through an extracellular matrix. And so the collagen is in orange, and then we'll see the cell in green. Play that. And so what this illustrates is that these neutrophils are able to sense and respond to a complex environment, and they have a goal. They're kind of migrating towards the wound site, and they're able to burrow and squeeze and kind of bend through this very dense mesh where the holes are actually quite a bit smaller than you would think the cell would be able to fit through. And so they don't have any eyes, there's no brain, and yet they can navigate this world just through a sense of touch. And so that's interesting, and we wonder how that works. And so so that's what we're working on here as part, helping Orion's lab kind of define the nanoscale environment in which we're kind of probing these mechanisms. And so so now at this point in the talk, Orion often likes to make an analogy that just kind of imagine if you were put yourself in the position of the cell. How do you solve this problem? So you have and he likes to mention this metaphor of a blindfolded rock climber. And so you imagine that you have like a rough idea of where you want to go. You want to climb up the wall. How would you get there? And then you'd have to solve several different problems that you would need to be able to sense where you are. So there's this a little video of an actual rock climber kind of sensing the environment. So you would reach your hand around and kind of probe and feel your way around where is a good place to grab onto. Next, you'd need to anchor yourself. So once you found a good spot to grab onto, then you would really need to kind of like pinch and hold onto that spot and brace yourself there. And then one of the things that I had never thought about, but is also remarkable, is that you'd also need to be able to release when you're moving on from a productive hold. As you crawl past it, you eventually need to release it. So it's pretty easy to bind something irreversibly, but it's a bit harder to let go of something once you're done. And this is something that has to happen like on a pretty fast time scale. So so we can kind of imagine what problems the cell has to solve at this nano length scale by putting ourselves in this position of the blindfolded rock climber. And so when we think about the cell, of course, it doesn't have a brain, there's no eyes, so it doesn't actually even have hands in the same way that our rock climber would. And so the cell has to solve these problems, but using nanoscale parts and tools that it self-assembles kind of on the fly. And so it builds its hand and senses the environment. It's able to anchor itself by kind of like wrapping around and polymerizing actin at the sites where it kind of grabs on. And then it's able to release those holds as it crawls away. And so understanding how the cell accomplishes this is really the focus of a lot of Orion's work, and then and our collaboration. And so that gives you some context of what we're trying to understand. The additional context of what we can measure is also important here. So our understanding of how cells sense and respond to shape in their environment is really limited by our ability to fabricate and control shape at the scale that the cells care about. And so, of course, you can just measure kind of spontaneous cell shapes. So you don't need any special nanopatterned devices for that. But then we can't really prove causality there. So we don't really, you don't have very much control, and you sort of just get whatever nature gives you. We can also use nanoscale beads, and so this is a figure from previous work from Orion's lab. And these are inexpensive and quite useful, but there's pretty limited geometric control. So we can see what cells do in response to kind of spherical structures, but if we want to go beyond that, and there's evidence that they seem to respond to saddle curvature, and you can't really make saddle curvature in a really precise way with a bead. So the third technology that Orion and his team has explored for inducing cellular curvature are lithographically nanopattern surfaces. And so using e-beam lithography, it's possible to build micro and even kind of nanoscale posts and channels and so on. But these are pretty expensive and difficult to fabricate, and the geometry is fairly limited. And one of the big challenges is just getting the cells to adhere to that shape. So we also need some kind of functionalization, some chemistry or something to attach the cells to these surfaces. And so that's another layer of challenge for that nano-patterning technology. So our collaboration kind of grew out of this need and interest in studying cells, but building sort of richer and more complex and sort of better controlled shape environments for the cells to probe and kind of measure the cells' response to their environment. And so we're calling these cellular playgrounds. So our lab focuses on DNA nanotechnology, and we think that these DNA nanostructures could actually serve as a really nice system for both defining very precise geometries and then also functionalizing them with chemistry or binding motifs that can also help the cells adhere to those geometries. So that's one of the goals, is just to develop DNA nanostructures that can address these challenges and provide them to Orion's lab so they can continue their studies on neutrophils. And then, but in order to do that, we have some additional challenges. So the types of shapes, the larger scale, and kind of the kind of complexity that we're encountering with these shapes that we want to build really requires a new generation of CAD tools. So there are some really nice examples in the literature of both DNA origami and DNA brick technologies that we'd like to adapt for the study, but we really need to go beyond what's been published and design our own shapes. And so that's the next challenge, is kind of designing a new generation of CAD tools for this project. And then that's a really hard problem, having developed CAD Nano and knowing what's involved with either building a new tool or extending an existing one. That seems like a really daunting task of just building new CAD tools. And so kind of the third goal of this collaboration is to work with Bret and his team at Dynamicland to begin to leverage Realtalk to build out these capabilities. And then we think we'll be able to go beyond what has been done with screen-based CAD tools. And yeah, we're really excited about how that's going to enable a lot of these construction of the cellular playgrounds moving forward. So I will return to what we've built a little later in the talk, but in the meantime, Bret will provide some history of the field of DNA nanotechnology and give us a sense of how it's possible to even build any of this stuff at all. Like where did this technology come from, and then how can it kind of feed into this project and enable what we're trying to accomplish.
So as Shawn said, we'd like to give you a history of the development of DNA nanotechnology, and we thought the best way of doing that is by taking you through some of the key papers that have been published, and not just telling you about the papers, but actually recreating the structures described in the paper here on the table. And so in order to do that, I want to introduce you to the little toy box that I'm going to be using here. I have a set of cards here with which I can play a little card game. For example, I can put this card down and then use this extend card to run up a strand of DNA. And then I can put it over here to give it a buddy, pair it up, and now we have a DNA helix. And these are still individual strands. I can grab this strand and kind of pull it apart and talk about it individually. I can pair it back up or pair it out of register. You can see these are not happy. But if we do have a nice helix, then we can see over here, in fact, a three-dimensional view of the helix. So this plastic tube here represents the helix, and then this represents the viewpoint. So it's as if you have your eye here and you're looking up at this helix, and then this is what you would see rendered in 3D. And so as I spin this around, you see that it's spinning around the helix that we defined. And so I can point to the nucleotides here in the schematic, and you can see that they're highlighting in the 3D view. But they're also numbered. It's either 1, 2, 3, 4, 5, 6. And these are the same numbers that you see here, 1, 2, 3, 4, 5, 6 in the 3D rendering. And they're also the same numbers that are actually physically printed on the tube. If you look at the tube, here's forward strand 1, 2, 3, 4, 5, 6. And this is really important because a lot of times it's hard to see what's going on in a 3D rendering, and you want to be able to look at the physical model because we're going to have a bunch of helices meeting in weird angles. And so to actually kind of build the model out on the table using plastic tubes, it makes a lot more sense as to where things actually physically are located in space. So the first landmark paper that we're going to take a look at was published by Alexander Rich, and he's not quite as generally well known as Watson and Crick, but he probably should be. He's kind of in that same cohort. He was born in 1924 and did a medical degree at Harvard before he decided that biochemists were having more fun and switched to that field. And he was in Linus Pauling's lab at Caltech when Watson and Crick published the structure of DNA here in 1953. And Pauling was also working on the structure of DNA at the time and kind of got scooped. So there was a lot of interest in that community at that time in nucleic acids. And Rich in particular became fascinated with RNA. So it was generally understood at the time that DNA had something to do with genetics, but nobody really had any idea what all that RNA was doing there and what it was for. And so Rich's big dream was to solve the mystery of RNA, which he largely did over the next 20 years or so, maybe culminating in his 1973 paper describing the structure of the transfer RNA. But back here in the 50s, nobody had any idea what's going on. So the first paper that we're going to take a look at here describes a pretty simple experiment that he did. So some folks had found an enzyme that when you put it together with a bunch of nucleosides, will then form those nucleosides into a long polymer. So if we say we have a tube with some adenines and uracils and put it in with this enzyme, then you get this polymer, which is just a random mix of A's and U's. And people looked at that, but it didn't really seem to be doing anything terribly interesting. And then Rich and some other people had the idea of, well, what if we made just adenines and just uracils, and we formed a poly-A, like that, and then separately we put that enzyme in with the uracils, and that would form it into this poly-U molecule. And so these started to look a little bit more interesting, but what Alex Rich did, and he says he doesn't remember why he did it and why nobody else did it first, was he took the poly-A and the poly-U and put them together. And simply by mixing these two things in the same tube, he said there resulted a very rapid increase in viscosity as well as a drop in optical density. From this viscous solution, tough glassy fibers can be drawn. And when you look at the diffraction pattern of those fibers, it looks like a double helix. So this was the first evidence that RNA could form a double helix. But more than that, this was the first evidence that you could form a double helix just by sticking two things together in a test tube without an enzyme. And that was completely astonishing because the only double helices that anybody knew about came out of the biological machinery, so they assumed it must be using enzymes. And also, single-strand nucleic acid in solution, I'm drawing it schematically as these rods, but that's not actually what they look like. They actually look like these balls of string. So how could two balls of string, both negatively charged, come together in such a way that they spontaneously form themselves into a perfect helix with perfect pairing? That was completely astonishing at the time, but that is in fact what happens. And so this is called the hybridization, and that is the core physical phenomenon underlying DNA nanotechnology. Now the next character in our story that we're going to take a look at is Ned Seaman. And he's kind of our main protagonist here. He's considered the founder of the field. He was born in 1945 and also started out as a pre-med and decided that biochemists were having more fun. He had a pretty miserable undergrad because he became interested in how biological molecules carry information, and his professors just kind of wanted to shove OChem down his throat. But here at grad school, he discovered crystallography and fell in love. And there was never any greater joy in Ned Siemens' life than solving small molecule structures using crystallography. It was just this dyed-in-the-wool crystallographer his entire life. And he went off to do a postdoc at MIT in Alex Rich's lab, where his first big paper was in fact describing the structure of this AU molecule that we modeled here. And he was also a co-author on the big transfer RNA paper. But in the later stages of his postdoc, he started getting disgruntled with crystallography. One reason was that it seemed kind of hit or miss, like either the molecule crystallizes or it doesn't, and you don't really have much control over the crystal. And also the fashion had changed from solving small molecules to solving proteins. And to Ned, it was like, there's thousands of proteins, you spend a couple of years on one, and then you go on to another one, and what kind of life is that? He wanted to do something a little bigger and more general than that. He ended up getting a position at essentially a non-research university where he didn't have students, he didn't have postdocs to work with him in the lab. So things were moving kind of slowly, but here in '78, a buddy of his asked him to take a look at holiday junctions. So those were described in 1964 by Robin Holliday as intermediates in genetic recombination. We're going to take a much closer look at those in a little bit. But that got Ned starting to think about branched DNA structures. And then in 1980, he had his big life epiphany, which was if you could use branched DNA structures to build a framework like this, a lattice work, then you could dock any ligand you wanted in each one of these cells, and you could just crystallize anything, right? So once you make this three-dimensional DNA crystal, this would be the crystal to end all crystals, and you would have just solved the crystallography problem in the most general way. So that was his big 1980 epiphany, and a couple years later, he published a paper, which is the next big paper that we're going to take a look at, where he talks about some of his ideas for how he could actually do this. So we normally think of DNA as a linear polymer. It's just one long string. And you can't build something like this out of just a single rod. You need joints, right? You need things to branch off. And so Ned says, well, what are circumstances in nature where you have DNA branching? Well, one of them happens during replication. So during replication, you get the situation where the two strands of DNA separate like this. And then you get strands kind of filling in like that. So during replication, this happens within a little bubble of the entire genome. But if you just look at this particular structure as it is, this is three helices meeting together at a point. And that's kind of what we want. We want three rods that meet together at a point. But the problem is this is unstable because this T pairs with this A, but this T can also pair with this A over there. And so what would happen is if you did happen to get this particular structure, these would actually come back together and kick these out. And so you'd end up with this longer thing all paired up like this, and maybe these would be over on the side paired with each other. So if you put these four strands together, you wouldn't get a branch, you'd get these two helices if you got anything at all. So this isn't quite going to work. So what else does nature have up its sleeve? Another branch structure that occurs in nature is the holiday junction. And so the way that works is if you have two homologous stretches of DNA like this, like these two, then you can have a strand that comes over like that, a strand that comes over like this, and they switch places like that. And so this is called a crossover. The strand is coming down this helix and then jumps over to the other helix and continues. And here we should actually be looking at this in 3 dimensions. So if I turn these the right way, then you can see that crossover right in there. That's this right here. And this is how Holliday drew them in his original paper, and this is actually kind of what they look like for real in solution. But the way that people normally draw Holliday junctions in their textbooks today is in a slightly different form that emphasizes that you actually have, or you can think of it as kind of 4 separate pieces of helix. And so I can rearrange this diagram by moving this helix over to here, and then moving this up over to here, and moving this helix around like this. I'm not changing the structure at all, I'm just rearranging the diagram. But this is in a form that emphasizes that it can be 4 helices that are meeting together at a point. So that looks promising. We want to build things like that. The problem is, again, that this is unstable because this T pairs with this A, but this T can also reach across and pair with this A. And likewise, this T pairs with this C, this T can also pair with that C. And so these can pair together, and then the junction will slip. Basically, these will go onto this helix, or these can come onto this helix. This junction can move around all over the place. And so Ned's first big idea was, well, we're synthesizing these things from scratch, so we don't actually need that symmetry. We can change it so that junction does not move around. So if we change this T right here to an A, and now this A can't pair with that, it can only pair with this T. This junction can't slip in that direction. And likewise, this C here, if we change that to a G, then it can't pair that way, it can only pair that way. And now we have what Ned called an immobile junction. So if you put these 4 strands together, and you've actually designed the sequences so they pair in this way, and there's not some other form that they prefer to pair, then you will actually get this cross-like structure. So that's great. What are we going to do with that? Well, for this next part, I'm going to shrink these down a little bit. So Ned's second big idea was that you could then form a lattice out of these. And the way that would work is using sticky ends. So if we extend this strand here with a couple of unpaired bases, and then have matching bases over here on this side, then if we had another one of these in the tube, which we do because we have a billion of them in the tube, then it would come along, and if these paired, then it would just kind of dock in right there, and we could get a big kind of infinite line of these things. And then if we want a 2-dimensional lattice, then we just do the same thing here. We extend the sticky end on that, which will pair with a sticky end on there. And then we can get another one here. So you can imagine this going out into a 2-dimensional lattice of these junctions. And then the last little bit to Ned's paper was, well, we want to make 3-dimensional structures, right? And the trick there is to realize that we've been working in 3D all along, because when we have these junctions here, the second helix can kind of come off the first one at any angle. And the angle that it comes off depends on which nucleotide the junction is at. By choosing how long we make these arms, we're choosing the angle that this helix comes off, and we can actually build structures coming off at an angle. We can build 3-dimensional structures. So that was the content of Ned's 1982 paper, Improvising Functions and Lattices, considered the seminal paper in the field, even though maybe it didn't really have much of an impact at the time, but it's, in retrospect, it's set the stage for everything to come. And this was a mostly theoretical paper. He actually did, I think, make an immobile junction a year or two later, but he really didn't get things started until he was able to move to a university that was an actual research university where he could attract students and postdocs who would actually do work in the lab with him. And then things started moving pretty quickly. So here in 1991, they published a cube a set of DNA strands that when you put them together, they form into this cube-like shape. This is not actually a cube because these joints aren't rigid. It kind of folds flat, but it is with a topology of a cube. It is the first closed shape ever made with DNA. Then a couple of years later, they published the double crossover. This was the first rigid structure made with DNA, and you can't build one of these without rigidity, so this is pretty key. So to show how that works, I'm gonna put this one back in its original form. So what we have here is called a parallel crossover. The strand is coming down this helix and hops over to the next helix and keeps going in the same direction. And Ned found that if you build structures out of parallel crossovers, they would tend to just fall apart. But something else that did seem to work is the anti-parallel crossover, where if we're coming up this strand, then we could cross over to the other helix to this strand and then come back down the opposite direction. And likewise, we're coming down this strand and then we could cross over to here and come back up. And so if we look over in the 3D view, you can see that these are actually crossing over, and the schematic kind of looks like these are going in parallel, but you can see here, these really are crossing over each other. And so if you build that, what you end up with is two helices that are tied together at a point where the crossover happens. They can still pivot like that around that point, but they are rigidly tied together. And so if we want to make this a rigid bundle of helices, then we need to tie it together at 2 points. And we can't do that just anywhere. We need to do it when the strand goes all the way around one turn or some multiple of turns. So if we look here, we can look at a 3D view and say we're crossing over here, and then we're going around the back side of the helix, and then about here, DNA is about 10.5 bases per turn. And so around here is where we could actually do another crossover. And I'm kind of running out of strand a little bit, so I'm going to extend these up a little bit more. So if we're coming down this strand, and then we cross over to here, and likewise coming up the strand, and then cross over to there. And my 3D renderer right now can't render loops, so I'm going to just add a little nick there for expository purposes. And now in the 3D view, you can see that we have two crossovers, one around here and one up around here, right? And so with this, we actually have two helices that are tied together at 2 points, and they are rigidly connected. And so in Ned's paper, he kind of looked at all possible combinations of crossovers of parallel, anti-parallel, odd, even, and he found this one, which I just demonstrated here, is the one that works, and it's the one that everyone's been using for the last few decades. So that was 1993, and then in 1995, Ned attended the first DNA computation conference, where he ran into a couple of young fellows from Caltech, Eric Winfrey and Paul Rodemund, who were interested in DNA computation. Their big dream was to make computers out of DNA. And Eric was in the middle of his PhD at the time. Paul had just graduated. And we'll come back to Paul in a little bit. But Eric had done some work with Stephen Wolfram. He was familiar with the model of computation that involved tiling out two-dimensional structures in a plane, and he saw what Ned had been talking about with two-dimensional lattices and thought that could be a good substrate for that form of DNA computation. So they collaborated on this paper where they made the first two-dimensional crystals out of DNA. And if you look what they did, it's basically everything that we've been talking about. You can see the anti-parallel crossovers, you see the sticky ends. It's just a lot of the ideas from Ned's original paper brought to fruition. in this paper. So those were two-dimensional crystals, and then 10 years later were the first three-dimensional crystals made out of DNA. And they don't really look like this. You can see that they're more triangular, but this is 3 very short, specially designed strands that when you put them together, they tile out into this huge crystal. This is, the cell is nanometers, and this crystal right here is 200 microns across. So it's this macro scale object where they know exactly where every single atom is in it. So this is a huge achievement and a bit of a fulfillment of the original dream of designing 3D crystals out of DNA. But it wasn't a fulfillment of the whole dream, because the original dream was that they could dock ligands into this crystal and then be able to solve those structures. And these triangles here are a little too small to put anything in. And they found when the triangles are bigger, the resolution went down. It wasn't really rigid enough. So Ned never quite achieved that dream of being able to make the crystal to end all crystals. But in trying to do so, he established all of the principles and concepts that you need to build stuff out of DNA, and other people took those concepts and ran with them. So the next person that we're going to talk about is Paul Ruman. He was a bio and CS double major at Caltech, did his PhD with Len Alderman, then went back to Caltech to hang out in Eric Winfrey's lab. And in 2006, he published the method of DNA origami. So that's the next big paper that we're going to take a look at. So all of Ned's work involved crystals, these perfectly symmetric, repetitive structures made out of small strands of DNA. And Paul's idea was to make arbitrary, asymmetric shapes by folding a long strand of DNA. And he built that on top of the same double crossover structure that we just saw. So if we imagine that we have a long strand of DNA, which say goes up this helix and then say hops over to this helix, comes back down, and we'll even make it into a loop. Again, I need to nick my loops so they show up. So these are 2 helices that are connected to each other, but we can keep on repeating this pattern. So now we have 4 helices, 1, 2, 3, 4, and we have this one strand which is coming up this helix, coming back down this helix, and we can have it cross over, then come back up this helix, cross over, come down this helix, come up here, and cross over to here. And so here's a single strand which is kind of going up and down all 4 of the helices. And that would give us a 4 helix bundle, which is tied together. And these 2 helices are only tied together at this point, but we could likewise cross over this pairing strand over here, and this would come around and cross over here. And so we have one long strand going through the entire design, and then these shorter strands which are pairing with it and providing the cross connections between the helices to make it rigid. And my scaffold in this particular example is only 80 bases long. Paul was working with about 7,000 bases from a bacteriophage that they found in the sewers of Munich. And so he was able to do more fancy things. And so by going up and down these helices in this raster pattern, you can form a star, you can form these triangles, you can form this classic disk with 3 holes. So Paul published this paper in 2006, but I think he'd been going around talking about it for a couple of years before that. And one of the young, impressionable people that heard him talk about it was Shawn Douglas. He was just starting his PhD when Paul was going around talking about it, doing his PhD in William Shee's lab in Harvard, and William Shee was also a new professor. And so they got very excited about origami and decided to just like focus entirely on origami. And so the lab, in a few years, published the method of how to do three-dimensional origami. And then also around this time, Shawn published CAD tools so you could draw these shapes on your computer screen. And so that led to a big surge of interest in origami because it meant that high schooler could design and fabricate a nanostructure in a day. So there's a big boost in accessibility. It also led to a big surge of interest in DNA and nanotechnology because it was suddenly so much more accessible and led to people looking for other architectures other than origami. And so that brings us to the last person in our story, Peng Yin, who also did his postdoc at Caltech with Eric Winfrey and Paul Rodeman, and then went off to the same lab at Harvard that William Shi was at, and his lab published the method of DNA bricks. So DNA bricks is a method for defining arbitrary three-dimensional shapes. And what they call a brick is a single strand of DNA, which is 32 long, with 16 going up one helix and 16 coming up another helix. So when it's put together with all of its buddies, it forms this kind of U-shape, which we can see over here in the 3D model. So these pair with other brick strands, and the way that works is you have another one of these U-shapes where the first half of it pairs like that, and then it comes over to another helix for the other half. And so this trick here is that because there's these 8 basis stretches, neighboring helices, such as this helix here, is forced to be at a 90-degree angle to these 2 helices. So this is kind of hard to see in this 3D view, which is why it's nice to have these plastic tubes here so you can see the L shape that these 3 helices are making with each other. So if you have a brick that is here, by putting a brick here, you kind of go north, and then by putting them at these other 4 corners, you can go south, east, west. You can fill out your 2D plane, and then by going up and down the helix as much as you want, that's your Z. And so each one of these represents a voxel in this voxel space. So Shawn's lab is mostly focused on DNA origami, but for this most recent project, they thought that DNA bricks would be more appropriate. So now I'm going to turn it back over to Shawn, and he's going to show the project that they're doing with DNA bricks.
We thought that the DNA brick method could be really useful for generating our cellular playgrounds because you have this cuboid set of voxels. So if you include every voxel or every DNA strand, you get the cube, but then you can leave out subsets of those DNA brick strands in order to approximate different shapes. So it's a very flexible method for getting a lot of mileage out of the same set of DNA strands. And so returning to this challenge of how do we define cellular playgrounds, that was the method that we wanted to bring into this collaboration with Orion. And so, and just to recap, we're interested in fabricating custom nanoscale shapes. We want to do it inexpensively in a way that's scalable and overcomes some of the challenges of both nanoscale beads in terms of expanding our architectures, and then also goes beyond nanopatterned lithographic surfaces so that are expensive and very low throughput. So now I'm going to talk a bit about the new generation of CAD tools that we've started working on in Realtalk. And all of our CAD tools are in this envelope right here. So I'll just pull this out and then come back to this later. So in this little zipper pouch, we have a voxel editor. So we have a front view of our voxel space, and we also, it's very easy to make multiple views. We can just create a top view as well, just by changing one line of code. So these are two views of the same voxel space. And then I have primitives here. So this program says that it's a cuboid primitive, and we have some code that defines what that means in our kit. And so what we see here when this is on the table is a whisker that sticks out 10 inches or so, and then we have a couple of different parameters here. So there's a depth parameter, height and width parameter, and then a z height. And so when I poke this into the voxel world here, we can see an actual design appears. And so right there, I can move it around, and I can use a little knob here in order to change these parameters. So for example, I can move the z height or z position around. We can make shapes that are smaller, have different dimensions, so I can change the depth a little bit, or here's increasing the height if I want it something that's long and skinny. I can just move this around and vary these different parameters here. To replicate the original paper, we can tune these parameters. But actually, we're not so interested in cubes exactly, but we are interested in other primitives. And so what we've been actually working with is not the cuboid, but we're thinking about our bead structure that we have, and that's kind of the starting point of the is polystyrene beads of a certain size, roughly 100 nanometers. And so our first thought was, can we just approximate shapes that we're already working with, as because then we have a positive control, because we can get signal, or Orion's team can get kind of a cellular response from a 100 nanometer bead. Can we make a 100 nanometer bead out of DNA bricks? And in principle we can, but that structure will be quite expensive. It's going to cost a lot of DNA. So we actually, instead of going straight for the $20,000 100 nanometer bead, we realized we could do a little trick here. We can actually kind of truncate the structure by dragging the whisker off kind of to the edge of the shape. And so here, this top down, you can still see that it's circular from the top, but then over here, it's kind of going off the edge of the world, the voxel world there. And so it makes a smaller structure. And I can pick up that structure over here in a tube. So I can go over here and grab that. So there's a shape, and it's a little bit hard to see from that angle, but I can use a laser pointer and start to tumble that shape a little bit. Okay, and so let me just set aside the editor for now, and we can look at what we can do with this shape. We dug into the supplementary material of the DNA Bricks paper and also the source code of the NanoBricks software that the Yin Lab published, and we re-implemented that in Realtalk, and so that's represented by this tool right here. And so what this does is it creates a box, and any voxel shape that you put in that box, it will translate that into a DNA brick design. So it goes from voxels to strands. And so here we have this hemisphere or half sphere shape, and we can place it here, and now we can see this tool is now offering a DNA strand design. And so we can use another empty tube to pick that up. And now we have a little bit of a construction history here. So this little tube is kind of holding some information that defines these strands. So we can tumble this around to see it, and then this is it's pointing back to the original design, so we can kind of keep track of which voxel shapes define which strand sets. And so, so this is the, actually this is the follow-up paper. We're using the architecture that's the follow-up paper to the original DNA bricks paper. So slightly different than what is shown in that paper. So once we have our strands, then it's just one more step to go to an order. So we're translating this list of DNA oligonucleotides, which we call DNA brick strands. Those have to actually be synthesized on we typically order these from a commercial vendor, and then they put these individual strands into 96 or 384 weld plates. And so if I put this inside the little design box here, then we get another we get the output of this plate order tool. And so now what we have on the table is some 384 weld plates, and we can see some information about the design that we just made. So we see this is almost 1,400 oligos and over 64,000 nucleotides total. And given our quote with our commercial vendor, then we expect that to be a little over $5,000 for that order. And so that's our plate order. And what we actually started with a design like this, and we ordered it, but then we also realized we might not need to fill the entire half sphere or hemisphere structure with DNA. So we kind of care about the outer shell of the shape, but we thought that we could save money and actually have some space for functionalization by hollowing out a cavity inside of this structure. And so in order to design that, we go back to our editor here and go back to our sphere primitive. And so again, we can get back to our original shape that we designed. But then we now use a separate primitive, which is a negative sphere. So this is just going to subtract a sphere from that design. And so what we can do is we take the original sphere, which has a radius of 10 voxels, and we can have our negative sphere, and we want it a little bit smaller. We don't want to subtract the entire sphere, so we can put our knob there and get it close to the radius, and maybe we shrink this radius down to 8. And so now we have a little bit of a smaller negative sphere, and we want these to be in the same Z as well. So there's that. And so now I can poke this in here and try to line up those the two whiskers. And so now it's a little bit difficult to see in the this box here, but we can pick this up in a tube, and maybe we want a little bit of a different position of the cavity, so I can grab this in another empty tube. And so we have a couple of additional designs here, and so we can tumble them in order to see what they look like. So, and what we want to do is actually tumble them so we can look underneath into the bottom here. So there's the first one and the second one. So we can see that still a little bit hard to see, but we have this hollowed out cavity, and so this one hollows out quite a bit. So we see there's only 1,334 voxels in that design, and then for the second one, I moved that negative sphere down a little bit, and so it's including more of those voxels in the design. And so one of the challenges in DNA nanostructure design is just managing all these different oligos. And so one thing that's really nice about this tool is that we can just arrange the structures that we want to order on the table, and then we get that order sort of auto-generated here, that we can pick this up in a tube and then ultimately translate that into a spreadsheet and send it to our commercial vendor. And so maybe I won't order this original structure, but I'll go back to my strand tool. So we're going to go from voxels into strands. So we'll pick this one up in another tube, and then let's get this one as well. We have another voxel shape here. Pick that one up. All right, and so we have strand versions of all of these different shapes. We can set aside our editor, and we actually can set aside our voxel shapes as well. So just focus on the strands here. So we're gonna, if we wanted to order all three of these, then we place all three of them in the plates to order tool. And so we pack them all in here, and then finally we can pick them up in another tube here. So that's our order, and not too bad. So we increased the size a little bit, so we're ordering the original one, so we have some additional shapes now. And so it's a little bit more expensive, but we now have 3 different shapes, and here we can see we're auto-generating the different pre-stocks as well. So I'm not going to show it here, but we can put this plate order into a separate tool that would generate pipetting instructions and a protocol in order to actually mix all these DNA strands together and fold them and assemble them into these shapes. So we now have our plate order, and then what we have been doing in the lab is we actually have ordered shapes that are very similar to these structures that I just demonstrated. And so I can set aside all these tools and go back into our notebook and show some of the preliminary data that we've been collecting in collaboration with Orion, and Mizuye has been collecting this data. Let's go back in here, and the last thing I'll show is just some of our early progress on this front. So we have unpublished data here, and so what we're looking at here are 4 different frames of live microscopy data. So we have neutrophils that we've added to different kind of playground environments. So we've put either polystyrene beads down on a glass slide, and those beads are labeled with a fluorophore, or we've put our DNA brick dome structures down. And so our domes are not quite 100 nanometers in diameter, they're 80 nanometers, but yes, similar in size to the polystyrene beads, but not a full sphere. And so what we were looking for is just do the cells respond in the same way that we expect them to when they crawl over a polystyrene bead, or maybe in a similar way. And so first we can look at some of the data for when we label I should say when Mizuye and Orion label the WASP protein with a green fluorescent marker, then what happens, and I'll play this movie here. So as the neutrophil, which we see in green, as it crawls over these beads, we see co-localization of the WASP protein. So these bright green spots show up at locations where the red beads are located. And we see some co-localization of both the beads and this green signal here. And so so that's kind of what we expect when the cell crawls over the polystyrene bead. Now with the wave, there's another protein called wave, or there's a wave complex, and this SRA1 protein in this case is now labeled in with a green fluorescent protein. So we can play this, and we see a little bit of a different response. So we're not seeing as much of a bright signal there, but we do see some co-localization of signal. It's just hard to perceive in this movie. So that's kind of the control data. And then the question that we had is when we put the DNA brick dome structures down on the slide and the cells crawl over them, do we see something similar? And so in the case of WASP, you can see here there's a pretty dense in DNA structures, but we also start to see some of these co-localization of these bright green spots within the cell. So not quite as bright, and maybe that's expected because these are smaller than the polystyrene beads, but nevertheless we were very excited to see this kind of response. And I can now also show the wave experiment, so where we have the other protein labeled, and again, kind of hard to see what's going on here, and so we had to resort to some statistical analysis. So we have so we collected all these data into histograms, and we can start to see in the WASP or wave case in blue and orange respectively, do we see like we can count how many beads or DNA origami structures that kind of light up and co-localize with positive signal. And so in the bead case, you can see WASP, there's many beads that are lighting up quite very brightly, whereas the wave are kind of not generating as much signal. And we start to see kind of a similar trend there with the WASP signal is co-localizing with these DNA dome structures. So that was really exciting for us to see that at least we could reproduce some expected behavior, and it was very encouraging for us to continue moving forward with additional designs. And so just to conclude, now what we're doing is thinking about how to zipper the cells to these DNA shapes. So we want to modify the DNA nanostructure design so they have single-stranded DNA handles, and then we will functionalize the cells with complementary single-stranded anchors, and then hopefully when those bind, they will really adhere to the DNA shape. We're also now looking beyond the dome and sphere structures into different types of curvature, different radii, and just really starting to probe some of the questions that have not been accessible with lithographic techniques. And eventually we would love to generate novel devices that are just completely unimaginable with lithographic patterning. So devices that might have different force responses in different directions, or that we could even use to kind of sense and generate forces when they're interacting with the cell. So now that we're enabled with these new design tools, we're really excited to explore this design space that has previously been pretty difficult to access with the existing tools. And so yeah, we're going to continue this collaboration and hopefully have more data to share in the near future.